Well, welcome everybody to the first Journal Club live streamed, courtesy of the Life Extension Advocacy Foundation, LEAF. My name is Oliver Medvedik, and I'm being recorded live streaming from the Cambar Lab for Biomedical Research, of which I'm the director of at Cooper Union, which is located in Lower Manhattan, city of New York. And that's my mess back there. So we'll be kicking off the Journal Club with an article by Ocampo et al. From the Belmonte Lab. And this is a Cell paper, and I'm going to share my screen with you so you can see the article. There we go. Share. And there. Okay, so hopefully that's coming in clearly. Let's move this around a little bit. Okay, very good. So the article is In Vivo Amelioration of Age-Associated Hallmarks by Partial Reprogramming. So what does that mean? So I really like this paper. It's really good, in my opinion, in that it sort of puts together correlation and causation of epigenetic reprogramming and how it pertains to aging, and that it's very strongly suggestive that both epigenetic reprogramming or epigenetic modifications correlate as well as are potentially causative agents behind many things that happen in aging. So what did they do in this paper? Well, just a summary of epigenetic modifications. The epigenome, as most of you probably already know, are heritable factors or heritable modifications that occur in a cell that are not directly due to the underlying ATGC code. So it could be heritable RNAs. In this case, these are modifications that happen to either DNA, in this paper, DNA or histones. So in this case, they're looking at the histone code. And here we see a summary of the paper on the very first page. Let me scroll here. And what you see in this box here is essentially an illustration of methylations on several histones, histone 4 and histone 3, H3 and H4, lysine 9 and lysine 20, that's the K9, K20, and there's a trimethylation there. And what this is summarizing is what happens in cellular senescence and DNA damage, in that you, if you have some of these changes occur, this correlates with cellular senescence, and by introducing 4 of these factors, which are OCT4, SOX2, KLF4, c-MYC, which are the Yamanaka factors, which were discovered to be the 4 necessary factors to genetically reprogram or epigenetically reprogram a cell into an induced pluripotent stem cell back in 2006. Nobel Prize winning work from the Yamanaka lab. These 4 factors, by reprogramming the epigenome, you can essentially ameliorate many of the effects of DNA damage and cellular senescence. So in this box as well, they show that, so this is basically a summary of the whole paper. And what this paper suggests is that if you take a progeroid mouse, so they have a mouse model that is suffering from an accelerated aging condition, we'll go into that in a little while, and you introduce these factors, and we'll go into how they introduce these factors in a little while, you can essentially rescue the mouse to some extent, meaning that you can extend its lifespan and give it a fairly youthful phenotype. And they also look at older wild-type mice, so not a progeroid mouse model, but a normally aged older mouse. And if they add these factors, they see improved regeneration of various tissues. And they look at pancreas and muscle. So let's take a look at the data in this paper and see if it bears out all of these suggestions. Okay. I'm scrolling through here, and I'm coming to the first figure. So I'm going to scroll through and look at the legend of the figure. Good. correlates with what I'm seeing on the screen here. So figure 1, amelioration of cellular markers of aging by short-term in vitro induction of OCT4, SOX2, KLF4, and c-MYC. So the mice that they use in this paper initially are these mice called Laki mice, L-A-K-I, and these mice have a silent mutation. It's a G608G, glycine to glycine silent mutation, meaning that one of the nucleotides is changed, but it's still a glycine. And this is a splice. It induces basically a splice variant. So it's a silent mutation that causes the lamin A gene to splice itself predominantly into the progerin version, which is a shortened version of lamin A, and it basically recapitulates, I believe, Hutchinson-Gilford progeria in mice. So it's a mouse progeroid model. And in these Laki mice, they further engineer these mice by having an inducible promoter that's inducible by an antibiotic, doxycycline, and the induction of this gene turns on the expression of four factors, and those are the Yamanaka factors, OSKM, OCT4, SOX2, KLF4, and c-MYC. And what they want to do essentially in this paper is see what happens if they induce these factors. Now, they do mention in an earlier paper that if you induce these factors too long or too much, that mice tend to form teratomas, and bad things happen. So the key to this paper is they wanted to work out a protocol in which they would induce these factors for just the right amount of time, so they don't get any negative consequences. So in this first figure, what they're looking at is tail tip fibroblasts. So they basically isolate cells from the tail of a mouse, these mice in particular, these engineered Laki mice, and they do a variety of experiments. And the data is presented here. So looking at figure 1a, they're essentially, these are basically like control experiments. They want to see if, okay, we add doxycycline. This is day 2 after adding doxycycline, day 4 after adding doxycycline. Do we see some of these Yamanaka factors get expressed or upregulated? So you could see immunofluorescence here in figure 1a, SOX2 and OCT4. The control is dark for OCT4, SOX2, I think it's low levels, and then there's emerging, those that I believe is DAPI staining for nuclei, and you could see the levels go up. So the doxycycline is expressing, causing induction of at least some of the factors from their gene. I'm not sure if it's an operon or if it's 4 separate promoters, but it's being induced. And figure 1b, what they're looking at is, do they have something that correlates with DNA damage? So this gamma H2AX is a histone that's associated with double-stranded breaks, and the control is just cells from the Laki mouse. So if you look at the box that has the green in it, the gamma H2AX, you see a lot of green speckles, noting that there's a lot of this histone factor that's being expressed due to double-stranded breaks, and this number goes down day 2 and day 4 adding doxycycline. So evidently the levels of gamma H2AX, which correlate with double-stranded breaks, go down, and the figure on the right of that is just a quantification of figure 1b, which you could see quite a big spread of foci per cell, so it's quite a bit of variability in the control, and it gets tighter and tighter and less foci with the treated cells. So that's pretty good. C is they do basically reverse transcriptase PCR, RT-PCR. So they just look at transcript levels of a variety of genes. Some of these correlate with P53 induction. Some of these correlate with like MMP13 and IL-6 with senescence. And you can see a difference in transcript levels. So you could see that factors which correlate with senescence, such as MMP13 and IL-6, go down. So this is 0 is day 0, then day 2, day 4. So they're showing that a lot of these factors that correlate with senescence, correlate with cells that are getting that are aged, where you see an increase of these various factors, they tend to decrease over time when you induce. So on to more controls. So let's see what happens to the cells. The histone methylation status. So in D and E, they look at, again, so this is all immunofluorescence. They're looking at histone 3, lysine 9 trimethylation status, and on E, histone 4, lysine 20 trimethylation status. And they're inversely related in that you have in aged cells or these lackey mouse cells, when cells age, you have a lowering of this trimethylation at histone 3, lysine 9, and you have an increase in histone 4 and lysine 20. And you can see the effect, looking at D, you can see that this gets rescued. You can see the number of foci of this methylated lysine 9 go up and inversely go down for lysine 4 K20. So what we have, all of these bar graphs and dot plots that you see on the right, these are all basically just quantifying the data that you see in these figures. So there's quite a bit of variability, but in general, the foci go down. Let's see, number of foci per cell. They go down for this one. They should go up for D. I don't know if I'm not reading this correctly. They do go up when you look at number of foci with these bar graphs. Scatter plots, yeah, I guess they go up. It looks a little bit more impressive than the immunofluorescence, though. But anyway, so that's suggestive of a correlation there. And then last, they look at this protein lamin AC. Now that's an antibody that they have against lamin A. So there's really 3 different versions of lamins, and I mentioned that these are progeroid mice, and so the progeroid version of lamin is progerin, which is a truncated version, but the cells can also express lamin A or C. I'm not sure if this antibody is for all 3 lamins. I'm assuming it is. So what you're seeing here in the control and then day 2 and day 3, these little arrows that you see pointing here, those are pointing to nuclei that have certain problems, which they call blebbing. So basically these little kind of bumps that stick out of the nuclei, and they're going to be quantifying that and showing that by day 4 and by day 2, I guess, as well, that these blebs, which are indicative of an aberrant nuclear architecture, go down when they add doxycycline and turn on these 4 factors. I find that interesting because these mice are making a defective lamin A. They're making progerin, yet the nuclear architecture seems to be improving. So I'm not exactly certain how that's happening. Maybe somebody could chime in that you have a nuclear architecture improving even though you have a defective lamin A gene. So I'm not sure what that says about what these 4 factors, these 4 Yamanaka factors, have with regards to progerin. I don't know if they're somehow shunting the RNA transcript to the splice variant to go more towards lamin A. I'm not sure, but it is an improvement. So you have less blebbing, or maybe somehow you're having less blebbing through the induction of something else that's rescuing the nuclear morphology here. Okay, so these are all short-term in vitro inductions, so only a few days. So again, obviously very suggestive here that you are having positive changes occur when you induce these factors. And then they move on to figure 2, which is Epigenetic remodeling during short-term in vitro induction of OCT4, SOX2, KLF4, and c-MYC, and how it reverses cellular markings of aging. So they're doing a bit longer-term inductions here. So if we look at figure 2, 2a, you can notice on the left side you have +4, -4, +4, followed by +4, -8, followed by +4, -4, followed by +4. That's just days induction versus non-induction. So +4 would be 4 days of doxycycline treatment, and then followed by 4 days of giving the cells a break, and then 4 days of doxycycline treatment again. And then 4-8 is 4 days of doxycycline treatment, followed by 8 days of no doxycycline treatment, and then analyzing the cells. And likewise for +4, and then -4, and then +4, which is just 4 days of dox treatment, and then analysis. So again, they're looking at, so these are Lackey cells still. So we're looking at these Lackey 4F TFs, these tail tip fibroblasts that they've removed from these mice. And they're doing immunofluorescence again. So again, you see OCT4, which is one of the Yamanaka factors. When you have a lot of induction here, +4, -4, +4, you have a lot of OCT4 being expressed. Those numbers tend to decrease quite a bit after only 4 days of not being induced. So, you know, it's a temporary induction of these factors. Yet we see some long-term changes here in gamma H2AX, which again is the histone that correlates with double-stranded breaks. In the control, which is the accelerated aging phenotype, you have a lot of these, you know, green speckles, which correlate to gamma H2AX accumulating at sites of double-stranded breaks, and then you have very little to varying degrees in the other treatments, and that they basically quantify as number of foci per cell, which is up here. Again, quite a lot of variability between cells, and this is just a bar graph version of what you saw up there, number of foci per cell, so that goes down. They do tend to go back up, and you can see the green speckles coming back up if you remove the doxycycline treatment and you let the cells kind of hang around for another 8 days. And then again, for the lamin AC staining, they don't have arrows here, but I'm assuming they're somehow picking out blebbing. So this is going to be the nuclear abnormal cells. So again, the number goes down, but it does pick up again if you stop dox treatment for at least 8 days. So in B, just like above, we're looking at histone 3, lysine 9 trimethylation status, and it goes up, which is the same as we've witnessed above, with only two days of treatment, or 2 days or 4 days of treatment. So this is, I'm not going to go belabor all this because we already went over what's on the y-axis here, or on the side here. But again, there's a correlation with an increase in trimethylation status, which is indicative of reversal of this particular indicator of aging in these progeroid cells. So these numbers go up, and we see in C and D, they're looking at a much more shorter term response. So we sort of had a mid-level response when we went up to, or mid-time, I guess, day 2 and day 4. Here they're looking at day 4 followed by a break, and then day 4 again. And here they're looking at shorter term 12-hour and 24-hour. So again, they're seeing the same responses in a very short-term effect. One thing that's interesting to me about the short-term effect is I don't know what the replication time of the cells are, but I'm assuming it's probably more than once. It's probably slower than once every 12 hours if it's a typical mammalian cell. If that's the case, then what we're looking at here is cells that are not haven't divided yet, but are still, you could still see a reversal of these effects that correlate with aging. So basically more methylation at histone 3 lysine 9 after 12 hours and after 24 hours, and also a decrease in gamma H2AX after 12 hours and after 24 hours. So this sort of suggests to me that, you know, these effects are not dependent on the cells dividing to sort of reset themselves, you know, for the next generation, but you immediately have some reversal of aging in the initial cell itself. So, you know, there may be significance to that. I'm certainly interested in, you know, how cells clear out damage through asymmetric division. So it's interesting if that is indeed the case, where an individual cell could repair itself without having to undergo division. You know, personally, I'd like to see a little bit more follow-up on C and D. I find that interesting. Okay, so on to figure 2E. So what are they doing here? I got to refresh myself with this inhibitor that they're adding. Oops. 4. Scroll down to, so there's so many figures here that they're all split up between different different pages. So figure E and F. So they're looking at a methyl transferase inhibitor, catechin, however you pronounce that. So it's an inhibitor of the methyltransferase that's responsible for the trimethylation of H3K9. So here's no inhibitor, here's with inhibitor. So they show that they inhibit this methyltransferase, and you could see lower levels of methylation. And they want to see if that correlates with lower levels of, or, you know, if this correlates with regulation of damage. So with inhibitor, you see more accumulation of gamma H2AX, suggesting there's more double-stranded breaks. You know, I guess there's a correlation here. I'm not sure if there's a causation. You know, you definitely, if you add this inhibitor of methyltransferase, you see less H3K9 trimethylation, and at the same time, you see more double-stranded breaks, as viewed by the accumulation of gamma H2AX. I can't remember if there's other data in the paper that suggests that this is actually, you know, a causal effect, but it's definitely a correlation. So, you know, the suggestion here is that, you know, that the, again, suggestion, it needs some follow-up experiments, that the epigenetic status is important for, you know, for perhaps for controlling DNA damage. Okay, that was figure 2. Okay, figure 2. How's everybody doing? Is there, let's see. Everybody doing all right?
We're all doing all right. You've got some chat I'm posting up.
Okay. Let's see. Are you, okay, is the chat coming up now?
Yeah. Do you see that some of the comments that I'm pasting into the chat on the right-hand side?
Oh yeah, here we go. See this chat. If the lamin A is modifying the nuclear architecture, maybe the role of the 4 factors is to modify the epigenetic structure into a form more suitable for pluripotency, i.e. the defective lamin A and the big 4 being opposing factors that pull the state of the nuclear architecture epigenetic state in different directions, says Matthew P. McTeer. Yeah, perhaps. If the lamin A is modifying the nuclear architecture, maybe the role of 4 factors is modified into a form more suitable for pluripotency. Yeah. Sam Moser. It seems to run on some form of science. Well, I take it you mark the parts of the genome you want to study with some of that glow-in-the-dark jellyfish DNA and see how it expresses. Sorry, I missed the start. Yeah. So basically, you know, basically are marking parts of the genome with glow-in-the-dark jellyfish DNA, more precisely fluorescent jellyfish DNA. But we're using, so we're doing, what we're using here are antibodies, so this is immunofluorescence. So these proteins are the H3K3M3, which is the trimethylated histone 3 at lysine 9. That's the native protein. And what you're basically doing here is you're incubating the cells with an antibody against this particular antigen, which is something that the antibody recognizes, and particularly, this epitope, which has trimethylation. And that antibody, if it's a primary antibody, is then labeled with another antibody, and that antibody to that first antibody has usually something tagged on it that allows you to see what you're tagging, and in that case that would most likely be the green fluorescence protein that's being tagged there, and for the OCT4 you would have red fluorescence protein. So so that's how they're visualizing that. Okay, figure 3. So I think, okay, so now we're moving out of the dish and we're moving into mice. So here's, here where things get more interesting.
Well, this is where it gets all sexy, yeah?
Yeah.
Sexy science.
Sexy old progeroid mice. Right. Okay. So they want to do this protocol, obviously, in mice. You can learn a lot about cells in dishes, but, you eventually want to see them in their natural habitat, which is going to be inside of an organism. How do they behave? So they want to optimize this protocol inside of mice. And remember, they had a problem earlier. You can't have too much of these factors, this OSKM, because too much of it and the cells basically completely de-differentiate, and you have these nasty tumors called teratomas, which are basically a composite of three different tissue types. So they look at a couple of factors. They have doxycycline treatment for a while, so not just 2 days or 4 days, but 8 days. So they have minus dox, plus dox, and then they look at body weight. And you can see that the body weight drops off quite a bit. Will lose 20% of their body weight in 8 days if you just keep dosing them with doxycycline. And the survival rate drops considerably. In only 8 days, you have quite a lot of die-off. This also, is a good example that you have to optimize these protocols, right? So you shouldn't be it's also a good thing to remind, young scientists that, just because you get a result you're not expecting doesn't mean it won't work. It just might mean that you haven't optimized the protocol. So I like the fact that they, hung in there and played around with the timing. So what they did was eventually come up with a protocol where they doxycycline treat the mice for 2 days, and then they don't do anything for the remaining 5 days. And then they look at transcript levels. So again, they're doing the same things they did to the cells that they derived from these mice that they did in the tail tip fibroblasts up in figures 1 and 2. They want to see if you get an expression of, of these Yamanaka factors over a period of time, and you do to varying degrees. OCT4 maybe a bit less. DOX, well, actually quite a lot. The DOX minus, there's nothing here, so I missed that. But yes, so you have quite a bit of expression of all 4 of these factors, and without DOX treatment, you don't. And here they show that, if you do this for a long time, it's bad. E is basically a post-mortem of a mouse that's got teratomas. These arrows are pointing to these tumors that have these 3 different cell types, the tissue types, ectoderm, mesoderm, endoderm, and that's indicative of a teratoma. So on to figure. So now they start kind of optimizing the protocol, and I got to scroll down to get to the legend here because it's all spread out. So they start looking at mice lackey mice that have been treated with dox for, for their optimized protocol, and they look at body weight. So it doesn't increase body weight. They still decrease. So lackey minus plus dox, lackey 4F minus plus dox. So it doesn't seem to improve body weight. It doesn't, make worsen it like they did before, but their modified protocol, doesn't improve it either. But what it does improve is survival, which is, of course, the most important thing. That's what we care about, is living healthy and living longer. So they seem to be living quite longer. So they're looking at age in weeks, and these are lackey mice that have no dox or dox. So this is the orange and the purple, and you can see they all drop dead around 22 weeks. Lackey 4F mice, interestingly, with no dox, you have a significant increase, not as much as with dox, but that suggests to me that perhaps the promoter is leaky. That's pretty common. People who do genetic systems, it's hard to have a really, really, really tight promoter, meaning that you put something in there and you don't have genes turned on whatsoever. So that's probably the explanation, but again, you'd have to do some more careful analysis to see if there's at least a little bit of transcript level of these factors in this case. But be that as it may, if you add dox, you get a significant, even more whopping increase. So you have the average lifespan increase quite a bit, from about 18 weeks to 24 weeks, and then the maximal shoot up to about 30 weeks. So overall, pretty impressive. And then they do look at various kind of gross morphology of the mice. C is just, pretty much as qualitative as you can get, minus dox, plus dox, wild type. So I guess what we can see here is that the plus dox mouse looks, these are age matched, is looking closer to wild type than the minus dox. So you could see the minus dox mouse looks a bit frazzled. It looks tired. Its legs are kicked out. Its ears are flopping over. It doesn't have a nice shiny coat. So they're, suggesting here that they look better. And after they sacrifice the mice, they look at, they look at the abdominal cavity, and they look at the gross morphology of the intestines and the other organs, and they show that the plus dox is, closer in appearance to wild type than this kind of, more nasty appearance of the minus dox prematurely aged mouse. And E, again, this is all, histology. So, this is for all you histologists out there. They look at skin, spleen, kidney, stomach, minus dox, plus dox, and for the skin, they show that minus dox, this is this dark staining is the layer of keratin. So keratinocytes, that's your outer skin layer. It's very thin, and with plus dox, it's got a thicker appearance, similar to wild type, and any other aberrations that are pointed out, spleen, kidney, stomach. Stomach is missing certain cells and it's less dense. You could see that it's getting rescued in the plus dox. And again, on the right, skin, spleen, and kidney, diameter of renal tubules, thickness of epidermis, this is all just quantification derived from these histological analyses. Same thing with the aorta. You have more nuclei here, suggesting more cells in the smooth muscle lining the aorta. And the heart rate. The heart rate is higher for plus dox versus minus dox. So those are just sort of the kind of the gross morphological, gross anatomical changes that positively correlate with plus doxycycline treatment. All right. Let's see. Let me move this aside here. Oops. Do we have any questions here? How's it going?
Oh, hang on.
Right.
We do have. Hang on. Let me post it to you.
Uh-huh.
And I've got a question as well. A point I wanted to mention was one of the criticisms that people have about this particular paper, and indeed a lot of papers, is the use of progenic mice. The classic, and you know what I'm going to say, don't you? The classic argument is, but that's not normal aging.
Mm-hmm.
What do you think to that?
Well, there's obviously a reason why these are used, and one main reason is that it makes results happen quicker. The other counter argument to using these types of mice is that there are correlations, with actual aging. So if you look at some of these, when they look at some of these factors later in the paper, such as the trimethylation of histone 3, lysine 9, that correlates, the changes that you see in these progeroid mice also occur in normally aged mice. All of this is to some extent artificial, even if you overexpress these Yamanaka factors in a wild-type mouse. The hope is, whenever you do any of these experiments, any model, like even using mice, for example, we really just want this to correlate to humans, right? So the hope is that whatever model system you're using is not going to be specific to just that model, or just that specific hyper-specific application, but that it's going to be broadly applicable. That's the hope. And you try to make sure as much as possible that that what you're seeing in your model at least correlates to a large extent what you're seeing in a quote-unquote normal system that you wish to then apply the results to. I know that's kind of a broad, kind of ranging answer that probably won't satisfactorily, satisfy everybody, but that's the best you can do.
Yeah, although I think the fact that they use, I won't say normal mice because they're not normal mice, but they use, you know what I mean, in terms of the experiment. They used progenic, but they also used, I'm trying to think of the word to use, regular mice, but obviously they were modified, so they would experience aging relatively normal.
Right, but you also have these issues, like there might be a leakiness of the promoter, and that might kind of somehow make them a little less normal. But then obviously you have to have control. So you would have a control mouse that's normal, right? That doesn't have anything in it, no Yamanaka factor induction whatsoever, no 4F Yamanaka factor system. So that's where controls play a role. So let's see. Sam Moser said, "Why did they choose fibroblasts? Probably convenience. They're easy to isolate. They're easy to grow. They're not. They can keep the mice around. It's not going. They don't need to sacrifice the mouse because they're taking it from a tail tip. Probably for those reasons." Let's see. Could deep learning algorithms be used to assist in your optimization process if they were fed enough data from your experiments to assist you in looking in the right directions? I don't know anything about deep learning algorithms. I'm just going to assume, sure, yes. There are definitely, pharma companies out there that are using these AI algorithms to help them optimize drug design. So yeah, sure. Or help you switch the right combination of genes on and off? Or is it purely trial and error, then refinement? Well, it's never purely trial and error. You obviously have to start off of something, right? You have to jump off of somebody's work somewhere. So that hunch is based on some data that's out there, usually. So how much trial and error you have is how much pure. So I would say it's never pure trial and error. Your results are already fantastic. Hypothetically, how much would that? Well, they're not my results, but they are pretty cool. Hypothetically, how much would that equate to human health span? Is it like an 80-year-old running a full marathon yet? Well, if it's this mouse study that you're looking at, I have no idea. That's again, it's really hard to again, like Steve mentioned, this is kind of an artificial system. Would that equate to human health span? Is it like an 80-year-old running a full marathon yet? Well, I think we've had 90-year-olds running full marathons, haven't we? Or was it like 5Ks? I mean, there are some people who can do that.
We've got a marathon runner in the UK, a Sikh gentleman, who I believe is 101, and he's from Birmingham, from where where I was brought up, and I think he's 101, and he still runs the Birmingham Marathon.
Okay, so I've got plenty of time to train and beat him. All right, good. Carla Parsons. So looking up doxycycline's type of antibiotic, I missed the very beginning. Yeah, it's just a system here. They're using the dox to switch on a gene, so it's basically an artificial system to act as a switch. It's very common for geneticists to make a system that's responsive to an antibiotic or something else that you can feed to an animal and use it as a switch. We can kind of go over that at the end of the paper again, just to kind of refresh ourselves as to how the system works. Okay, so now they're looking at Figure 5, amelioration of cellular markers of aging, again, by in vivo induction of OCT4, SOX2, KLF4, and c-MYC. So these are lackey for F mice, and I believe, let me see if this is already human on the bottom here. No, not human yet. That's the problem. They have these figure legends spread out all over this paper here. Okay. So again, this is from the lackey 4 mice. So this is in vivo. So just like I mentioned before, figure 4 and 5 is really just a repeat of what's going on with what they did with figure 1 and 2, which is basically, showing that these factors are being expressed in various cell types. So in figure 1 and 2, it was done primarily on mouse, the tail tip fibroblasts. Here they're looking at transcript levels in probably biopsy punches from various organs. And you could see the levels of these factors go up, they correlate. In B, they're looking at this protein, Ki67, which is just an indicator of proliferating cells. So basically cells that are dividing. So the more dividing cells, the healthier, I guess, the organ is, and the more potential it has for regeneration, assuming it's not a tumor you're looking at. So here they have wild type, and then minus dox and plus dox, and you could see more of these little punctate foci, which have been stained immunohistochemically using an antibody, showing that you have more Ki positive cells. Although it looks like there's more in skin, although maybe I'm reading this incorrectly. Let's see, plus dox, wild type, yeah. So stomach. Actually, wait, hold on a second. Kidney, Ki67 positive cells. So are they actually going down? I thought Ki67 was indicative of replicating cells. Shouldn't that go up? It's been a while since I looked at this, and it's getting hot in this lab. I thought it should have been the other way around, but again, maybe I just need another coffee here. I'll go back to that figure in a little while. So they're looking here at, again, the trimethylation status of the same histones that we looked at previously. They're showing it kidney, spleen, and kidney and spleen. They're showing that with dox, you have a recapitulation of what occurs in wild type. And let's see, wild type are these wild type, or are these wild type lackey cells? Lackey for F mice. I I think these are wild type not aged, right? C, let me double check that. C5C, make sure that these are not, it gets a little confusing sometimes when they say wild type. Does it mean wild type aged, wild type not aged? So let me go up here and take a look. 5B, wild type levels. Wild type levels. Is this wild type normal or wild type old or wild type young? I'm going to assume it's wild type young because I'm looking at that mouse up there. That's wild type young. Okay, so the numbers, yeah, okay, so I wasn't confused. The numbers do go up, but not quite as high as wild type. So plus doxycycline, you have more of these punctate foci that go up, corresponding to increased replicating cells. And again, this data here just recapitulates again what we saw in mouse tail tip fibroblasts. E is staining for this factor called beta-galactosidase, and that's like a classical marker of senescent cells. So it's expressed in cells when it should not be expressed, and it was kind of one of the earliest biomarkers that have been correlated with senescence. And you can see with no doxycycline in these lackey mice, you have a lot of senescent cells. So again, this is one of these correlations that is, again, suggests that what we're seeing in progeria, at least in this model, is, there's a lot of overlap with normal senescence, normal aging, because you see also more senescent cells in normally aged individuals. And with plus doxycycline, when you express those four factors, you have a kind of resetting happening. And here they're looking at skin or muscle on the left, skin on the right, I believe. PAX7 is a protein that is expressed in what are known as muscle satellite cells. And these are basically multipotent stem cells. So these are muscle stem cells. So if you have more muscle stem cells, you have more ability to regenerate, and that's correlates with healthier tissue. So wild type, you have a lot of these punctate foci. Lamin is just an extracellular matrix. So this laminin is not to be confused with lamin A. Laminin is part of the ECM, which is found between cells. DAPI is a nuclear stain. So what we really care about is this PAX7, and you can see more of these PAX7 stained cells go up, suggesting you have more cells appearing in doxycycline treatment that are muscle satellite cells. And cytokeratin 15 is, it's a keratin protein, which is a structural protein. I think it's indicative of healthy skin. That may have something to do with the skin thickness we saw earlier. And again, you see an increase in figure 5G. So based on all these cellular markers, and also the extension of lifespan in these lackey mice, everything is pretty positive so far. And that's getting us to our next to final image, which is next to final figure, which is amelioration of aging hallmarks in wild-type mice and human cells by short-term induction, in vitro induction of OCT4, SOX2, KLF4, and c-MYC. So now they're dipping their toes into normal mice and human cells. But in both these cases, this is still cells in a dish. So we're not talking about wild-type mice, and we're not talking about truly wild-type mice, as Steve mentioned earlier. We're talking about wild-type mice that are these 4F mice that have those four factors under control of a doxycycline-inducible promoter, and they're looking at tail-tip fibroblasts, just like they looked at with the lackey mice. And human cells, these, I believe they have some human cells here that they also are, I'm not mistaken. Let me go down to the figure legend. Wild type 4F cells. So they have some human cells that some human cell lines that they generated that also have doxycycline-inducible 4F region, so they can express those 4 Yamanaka factors. So those are late passage cells, and that just means that, again, late passage just means every time cells grow in a dish, they basically crowd, and then you split them. By splitting, you basically take some cells out and transfer them through another dish and another dish. And if the cells are not expressing telomerase, they undergo replicative senescence. So if you were to take fibroblasts from a biopsy as a primary culture, as primary cells, and then you would start a cell line, meaning that you would keep passing those cells over and over again. At a certain point, they would stop dividing, and they would undergo senescence. So these are late passage cells. So these are cells that are getting close to senescence. Okay. Any questions here? Let's see. Oh, we have some more statements here, comments, questions. Comment from Sam. My dad can. He's 64 and cycles like a ninja. All right, awesome. Is it worth exploring what allows that to happen? Sure is. I think there's a lot of people that are, a lot of scientists that are looking at genotypes and how certain genotypes, certainly from centenarians and supercentenarians, the type of people hopefully that your dad will become, which is people that can basically run marathons at 80, and what is it that allows them to basically, make it to a very advanced age and still be very healthy. So yeah, people, there's labs that are certainly looking at those differences, and there have been already some kind of intriguing correlations with various mutations. Or could it be something similar to what you're trying to achieve occurring naturally? Has a sequencing project been attempted for these types of individual large scale? Yeah. So I mentioned there is. I don't know of a specific one off the top of my head. I know there's been publications, and we can certainly pull up those papers. And I previously asked, use machine learning to literally spot the difference between these super fit old folks, and see no one to pinpoint their differences and identify if it's a combination of genetics and environmental factors. Yes, so that's precisely what they're doing. So obviously, just looking at the sequences, by eye as a human being, it would be way too tedious. So there are definitely people that are designing algorithms to sift through all that data and make correlations. So that's not my expertise, but there are definitely computer scientists on board that are helping out with that. So I wouldn't be surprised if... what's that Google group out there? What's it called again? I forgot. Steve, you know the name of it.
Google. Oh, Calico.
Calico. Maybe they're doing something like that with all their computational resources, but who knows?
I suspect they probably are. And obviously that's opposite to the damage repair approach of, say, SENS, for instance, where they don't put much stead in looking for these longevity genes.
Yeah, I think a multi-pronged approach right now is is something that nobody really knows where some of the key breakthroughs. I have my favorite types of research. I mentioned earlier I'm really keen on damage removal and segregation of damaged factors and how that happens in somatic cells in your body. Because after all, cells do regenerate. We know that. It's been happening for, basically forever. When a sperm and egg meet, you have a child that resets at 0, right? It doesn't take on the age of the parents. So clearly there's something happening in the germline that is not happening with somatic cells. Okay, so...
although this does touch upon that, really, in many ways.
It does, because of epigenetic reprogramming. That is correct. So, let's see. Amelioration, aging. Okay, so wild-type mice and human cells. Okay, so now they're into wild-type mice. And again, these are basically the same controls that they did in the first figure, but now they're applying it to wild-type cells. So you see how, I like how they're methodical here. You really you can't take anything for granted. You can't just say, well, it worked in our lackey mice, so it's going to work in our normal cells. And if it worked in our normal cells, it's going to work inside the mouse. And if it worked inside the mouse, of course it's going to work in humans. Well, you have to actually do all of those individual experiments, because you don't know, right? I mean, you literally don't know. You can have a completely different result in human cells versus mouse cells, and that could be telling you something important. Or it could be the identical result, and that's telling you something important as well. But you can't assume. You have to do the actual experiment. If you assume, that's just your hypothesis. All right, could be a good hypothesis, but you still have to do the experiment. Okay, so A, so they're doing the same control. SOX2 levels go up, OCT4 levels go up. This is with treatment with doxycycline. 8 gamma H2AX levels go down, meaning the damage, the correlation with double-stranded breaks goes down, and they have got the number of foci on the right. And also factors that correlate with senescence, such as P16, P21, go down. MMP13. And in D, they're also looking at the trimethylation of H3K9, and that goes up as it's expected. So what do we see here? Number of foci. I don't know why the number of foci is. So total intensity goes up, but the number of foci stays the same. Is that the same result that they had before? Or the number of foci goes slightly up? It looks like there's more foci. I mean, I don't. Here it says that they look identical, although the intensity, meaning that you have more trimethylation occurring at these spots. Let me go up to the other experiment and see what they saw there. Let's see where they counted foci. That's way back here. Okay. All the way at number 2, I think. Oh, yeah. Yeah, okay. Well, the number of foci look like they go up here. Well, wait, this is volume of foci. Number of foci go up, and the volume of foci go up. And this is short term, and long term. Long term is, all right, the number of foci go up slightly, I think. Total intensity is the same. All right, so there's a bit of variability there. Let's scroll down. Back to our figure 6. Scroll, 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 scroll. Okay. And E and F are interesting in that these are now, we're changing species. They are now looking at human cells. I'm not sure which human cells, maybe they're fibroblasts. I'll take a look later, but suffice it to say they're human cells. And they're looking at, again, OCT4 gamma H2AX, and it's very similar results to what we saw with the mouse cells. So these are human cells that are 4F, doxycycline treated. Again, the same thing with histone 3, lysine 9, trimethylation. The same correlations they see with number of foci and volume of foci and total intensity, suggesting that you have less DNA damage and correlating with more methylation at histone 3, lysine 9. They do have these Yamanaka factors. People have been studying them for years, so maybe this was already done in another paper, maybe it's in the supplemental. But again, if you really, really want to look at a system as close to reality as possible, then you want to minimize as many variables as possible. And one variable to eliminate is obviously this 4F gene, that was inserted to internally express factors. So it would be interesting to see if we can recapitulate all of this stuff by just adding in the factors externally without having something inside the genome. But that's like an obvious next experiment, or maybe it's already been done. Okay, so what they do in figure 7 is a little different. Is they used aged wild-type animals, and they see if they can have, if these treatments could actually help them resist metabolic disease like diabetes. Although this is a model, again, a model of diabetes. They don't really, it's not a diabetic mouse. They do pancreatic injury using this chemical compound called STZ, which is toxic to beta islet cells, and they injure skeletal muscle by using this toxic compound called CTX injections. And then so these are wild type mice that have the Yamanaka factors under the control of doxycycline inducible promoter, and basically this is the procedure here. You have a 12-month-old mouse that's it's genetically modified. You add dox for 3 weeks, then you damage either the pancreas, or, in this case the pancreas with STZ. You wait a week. So you add in STZ for a week, then you wait 2 weeks, and then you do this is a glucose test and histology to see how responsive the mouse is to glucose levels in the blood. So what they show in B is that if you have these mice and with no dox, you have higher blood glucose levels over a period of time, and with dox you have lower glucose levels. So this is when they inject the mouse with glucose, they see a spike in blood glucose levels, and then a normal mouse clears the glucose much more rapidly. It would be interesting. So this is minus dox, this is plus dox. So this is after this treatment. I would be interested to see what the baseline is before STZ. I'm not sure if they have it here. So that would be like a total control. They don't seem to have that there, but nevertheless, you see a decrease with dox treatment. And in C, they look at the amount of, after they add doxycycline, they stain for insulin. This looks like a cross-section of the pancreas. DAPI is nuclei. So these are beta islets, and those are little cells in the beta islets. So you have more beta islets and more cells when you add doxycycline after this treatment. So it appears that you have more pancreatic beta islet cells, after you add the dox. Although, again, how they came to be, I'm not sure. Probably cells redifferentiated, but there's not much more info that you can pick up out of this. And they look at recovery after muscle damage. Again, these are aged mice, meaning they've been sitting around. They're 12 months old, so I guess that's middle age, post-middle age for mice. Again, doxycycline. In this case, you now inject CTX, which is a toxin, to a particular muscle. They mentioned in the paper somewhere. You wait 10 days, you do histology, which is, sacrifice the animal, take cross-sections. And figure E is just showing you how this looks. So we have PBS, phosphate buffered saline. That's a control. So that's basically the carrier for CTX. So it's just like, water or something like that. So this is your 12-month wild-type 4F mouse without doxycycline treatment, 12-month wild-type 4F mouse with doxycycline. PBS. This is normal. You add CTX. I think what this is supposed to show is less density. I think all of this stuff is supposed to be, you have muscle cell die-off, and this is supposed to be denser. And this laminin is just, again, minus dox plus dox is just the extracellular matrix that surrounds the cells, so you have more of it showing that there's more cells. And they look at cross-sectional area. So the cross-sectional area of muscles, there's more of it per cross-sectional area. So with doxycycline treatment, so, the muscles are thicker, and you have less central nucleated fibers, which I had to look up, honestly, because I'm not a muscle expert, but this is indicative of aberrant muscles. Muscles normally have nuclei that are off to the side, so this is indicative of myoplasia or some sort of degeneration of muscles. So it seems to improve muscles. And again, here they look at PAX7. As I mentioned earlier, that's a marker that's expressed in muscle satellite cells. So when it's kind of hard to see here, but this is without DOX, this is with DOX treatment, laminin, which is the surrounding extracellular matrix, merge, little blue dots are basically nuclei, the little green dots here are PAX7, and according to them, if they count, you have more PAX7 positive cells. So with these, this last figure, it's sort of, not as, I don't think it's as weighty as all the other figures. There are more figures in the supplemental that sort of, back up everything we've said here. The one crucial experiment, obviously, is, okay, do these wild type 4F mice actually live longer, right? You have, obviously, people have been using these for some time because you have 12-month-old wild type 4F mice. I'd really like to see what happens to a 12-month wild type 4F mouse when it's treated with doxycycline periodically. Perhaps they're doing the experiment now. I don't think I saw it mentioned in the discussion. To me, that's clearly the most obvious next experiment. Clearly, it's going to take a while, and it's going to be expensive because you're going to have to be rearing mice. And, it probably has taken them this much time to kind of optimize the protocol using these lackey mice, right? Because you have to get the amount of doxycycline and expression just right. Otherwise, the mice will drop dead of tumors. Okay. James Joyce, when will the gene therapy delivery problem be solved? Like, if you wanted to express these somatic, isn't delivery still an unsolved problem? Yeah, it is a problem. Delivery, is a problem. You have to, there's a couple of problems for gene therapy. One of them is getting the gene to the right place. And that's kind of partially solved. So with the CRISPR system, one part of that equation is making a break at the right locus at DNA. And that supposedly is very, very, very efficient now. What's still not so efficient is actually copying in a gene into that location. So depending on the cell type, it's still fairly low efficiency. But, I actually have a project I'm working on to try to improve that efficiency to a much higher level by boosting homologous recombination rates. But the other main problem, the third problem is, like you mentioned it, delivery. You can get things delivered into, I guess, they're easy things to target and hard things, right? So brain would be pretty hard. Blood cells would be pretty easy, right? So if you take them out of if you harvest your hematopoietic multipotent stem cells, then you can fiddle with them in addition, put them back. It's kind of hard to do that with neurons. So, and it depends on, what sort of problem you have. If it's something that is only affecting one organ that's easy to get to, like white blood cells, then that's a lot easier problem to tackle with delivery than something that is affecting your entire body, and you need to actually, like if progeria, perhaps, the lamin A mutation is a problem for every single, somatic stem cell in your body. So if that's the case, yeah, how are you going to get, a repaired version into every single stem cell in your body efficiently? So that is a challenge, to say the least. Okay, well, I think that wraps the paper up. Any questions that are still out there on the Ocampo paper? Any comments and thoughts?
I'm just having a look.
Oh yeah, so just to go back to just the way that these mice were made, just to recapitulate, these were, somebody mentioned the doxycycline earlier. Yes, that's an antibiotic. And just to reiterate again, usually what researchers do is they use a chemical compound that's water soluble that a mouse could easily drink, and that compound can then enter cells pretty readily, and it can then bind to an activation factor, a protein that when it binds to that antibiotic or some other small chemical, it will change conformation and then bind to a promoter, like a gene switch, and then switch on a gene. So it's basically you use that chemical as an on-off switch. And so you're not using it as an antibiotic, but you're using it as a way to switch a gene on, and then you can have that promoter control anything you want. So in this case, they're having it control those 4 factors, the expression of those 4 factors. And potentially, depending on what sort of, I don't know if this is possible with doxycycline, but in some cases you can actually titrate the dosage. So you can have higher or lower levels of factors being expressed from a promoter. I'm not sure if that's the case here. I think this is like an all or none switch, so they can kind of only switch it on fully or off fully by adding it to the mouse's water.
Yeah, and that's really the barrier really here is to translation to humans. We have to find ways of switching these factors on, because obviously we can't engineer somebody who's already middle-aged. So there's a pressing urgency really, because if this works and it's looking good, we need to find ways to activate those factors. I don't know how you go about it, but I'd like to know.
Yeah. I mean, the most direct way, which is again still a challenge, is if you ablate your senescent stem cells using, using current drugs that everybody's looking into now, what are senolytics, then you can put back, rejuvenated stem cells, but you somehow have to get them back into the right location. I don't know if you can engineer like an Uber stem cell that can go anywhere it needs to go. But if you had something like that, you'd have to be careful that it doesn't turn into, the world's most powerful cancer.
Well, it'd be totally potent stem cells or pluripotent. But as you say, the risk with using totipotent or pluripotent is cancer. Even multipotent are pretty dodgy. Although they did recently create blood stem cells, which is very interesting because they're multipotent.
Yeah. So, we're not so, we're not early days. I've been in this field for, since about the year 2000 when I started my graduate program at the Sinclair Lab. And I'm running now a biomedical engineering lab for undergraduates here at Cooper Union, so I'm not doing aging research directly, although one of the projects I'm working on could certainly be applicable as a technology. But I have been keeping up with the literature, and I have to say it's been progressing quite a bit, and I'm pretty pleased at everything that's been coming out. I mean, as far as like to do an aging analogy for the field, it's probably in its adolescence right now, maybe young adulthood.
Oh yeah, and we got a question from Carla in the chat, and she's also said, well, maybe for next time, could we talk about the blood stem cell paper?
Sure. Yeah, why not? Carla also wants to go over the Yamanaka factors for anyone who doesn't know. So the Yamanaka factor paper is interesting. It was sort of a brute force approach, and basically they looked through the literature and looked at every factor that everybody was studying. I think they looked at like 20 different factors that had anything to do with getting cells to be induced into a stem cell-like state, and they kind of whittled it down very tediously down to at least 4 factors, and these are transcription factors. At least I think they're mostly transcription factors. that kind of act as sort of master regulators, in that they turn on genes that turn on other genes kind of in a cascade effect, and can essentially de-differentiate a cell, right? So they'll switch on other genes, which then set the whole process in motion that will cause epigenetic reprogramming to take place throughout the nucleus of a cell, and cause it to, basically, de-differentiate, turn a cell that's a neuron, for example, back into a stem or stem cell-like multipotent or pluripotent stem cell.
Well, they did that with very recently with dopamine producing neurons. They managed to get astrocytes, and again, it's a similar sort of thing. They used this was in a living animal, and they managed to use reprogramming just the same as this, or very very similar, and in a living animal, they managed to reprogram astrocytes, which are a very common cell, I understand, in the brain, and they managed to make them more sort of malleable, and then they guided them down the differentiation pathway, and they created dopamine-producing neurons. And within 5 weeks, they observed that dopamine-producing neurons had increased dramatically in the target animal, which is very interesting because obviously it's Parkinson's.
Yeah, that is very interesting. One thing still about stem cells is that it's still, again, fairly early. We know a lot about stem cells, but but there are different categories of stem cells, right? So there's the totipotent stem cells, which are something you get from early embryo, the first few divisions or the first division, which can basically just means it'll turn into any cell that you want. And then there's pluripotent stem cells, which can turn into, I forgot the exact definition, but can turn to any of the 3 different tissue types, mesoderm, ectoderm, endoderm. And then there's multipotent stem cells, which are also referred to as adult stem cells, which are the stem cells that are in various organs of your body, but can only turn into, specific cells within a specific tissue type. For example, your hematopoietic multipotent stem cells, I think they used to be called hematopoietic pluripotent stem cells, but just redefining it to multipotent stem cells, because these cells can only turn into every cell in your blood, but they can't turn normally into a neuron, for example. They'll turn into different types of white blood cells, erythrocytes, et cetera, T cells, B cells, but they can't, but they normally, that's kind of the limited lineage that they have. And there's a lot of big open questions in stem cell biology, like how many different multipotent stem cells do you really have in your body, right? Like, I think Wikipedia says you have like over 200 different cell types, and I'm assuming that is counting differentiated cells. So, is there 1 or 2 different multipotent stem cells for every different organ or tissue type, right? I don't know. How many different multipotent stem cells are there? Also, the whole issue with reprogramming cells, how well is this reprogramming working, right? So is it obviously it's going to recapitulate a lot of things that a normal multipotent stem cell will be doing, but do we really have it reprogrammed 100%, or is there, is it still a little off or a little wonky? That's always the issue. The other thing is, do we know how to differentiate exactly, the different states of stemness? That's still kind of being worked out. There's still fuzzy edges, right? So how does a cell, what are the degrees of stemness from multipotent to pluripotent? Right now it's just a big checklist, right? If it looks like a stem cell, acts like a stem cell, quacks like a stem cell, it's probably a stem cell. So, right now there's just a giant checklist. So if you have a pluripotent stem cell, one of the gold standards is that it forms the 3 different tissue types when you inject it into a immunologically compromised mouse, and it forms teratomas, right? So showing you that it can form all these 3 different types. So they're, I guess that works as an assay. There's a lot of other assays, obviously, epigenetic status, but there are a lot of fuzzy edges still, I think, in stem cell science, as to what are the exact delineations between different stem cells, and it'll be interesting to see how this all pans out. Okay, let's see, Carla Parsons, the Yamanaka factors. Is that the idea that young blood can de-age you? Using stem cells mean I don't have to vampire my kids while they sleep? No, I'm afraid you're still going to have to vampire your kids, but just make sure that you share the right blood type. No, just kidding. Don't do that. Well, we don't really know what are all the factors that are required, in this parabiosis that you're referring to. It does work. Obviously, you can get factors in youthful blood that would help regenerate older muscle in the aged animal. There's been some controversy in the past as to some of these factors supposedly have been identified, but then later the results couldn't be repeated. So I don't think it's really clear as to if specific factors have been conclusively identified. What I do think has been demonstrated is I don't think there's a transfer of stem cells, young stem cells, from the blood of a mouse to an old mouse. I think I have to pull up the experiments, but I think there were some experiments done where they had GFP labeled cells from one mouse and with a young mouse and an old mouse, and you could not see any transfer of green fluorescence marked cells from the young mouse to the old mouse. Although I could be wrong about that, but I think that was, again, I have to find the original paper. So it's clearly some kind of circulating factors. I think the distinction is that totipotent includes all cell types, yep, including the placenta, and pluripotent, any cell type from 3 germ layers. Yes, I believe you are right, Matthew, that totipotent, all cell types, including the placenta, or as now I remember in mice, the epiblast, and pluripotent, any cell type from 3 germ layers. Okay. Well,
probably dilution, by the way, about the blood. The recent paper by Irina Comboy, who I'm sure you've been following, she's spent decades looking at these factors, but she published a paper earlier this year that very strongly suggests that it's dilution. So it's not so much that the young blood has got useful factors in it that make you young, it's the fact that you're diluting the aged, pro-aging, pro-inflammatory factors in the body. So you've got things like IL-6, TGF-beta, you've got TNF-alpha, all these cytokines.
That is actually more promising, because you can come up with an aging antitoxin, right?
Well, what they're actually doing is they're proposing to use plasmaspheris, which is kind of like basically like dialysis, and what they want to do is effectively repurpose plasmaspheris and actually create a blood scrubber, and that's what they're doing right now at UC Berkeley.
Right.
Because they believe that only about 6 or 7 factors ultimately control the altered signaling environment, TGF-beta and some of the other things that they've looked at, and they believe that, and they did an experiment where they actually filtered these out and got more profound results in comparison to young blood, and they're saying it's dilution. We could talk about that paper as well, because that's really important. But that's what they're doing, and they're saying it's more of a case of what you remove than what you put in. So they think that they can scrub your blood clean.
Yeah, it does make sense. Maybe you could also come up with antibodies against some of these factors and have your white blood cells scrub them out internally, assuming that they still function at a high enough rate. I think all of these therapies are going to certainly play a role. I think this, what Steve just mentioned, all of this stuff is going to hopefully make us live longer and healthier. What I don't think fundamentally is it's going to fundamentally, all of these therapies are going to fundamentally push us past our supposed natural maximum lifespan of 125 years, give or take. What I do think is going to happen is that this will allow more of us to reach that age in a much more healthier state, which is a pretty damn good thing. But I think, just like with Steve, I'm also interested in like really the fundamental mechanisms of aging, which happen, it's clear to me at a cellular level. So if you look at individual cells, even organisms that are acellular, like yeast or bacteria, they still, will age if you track them. So there's something happening at the cellular level that is still, causing cells to hit a wall, to accumulate damage. And I think personally, until you tackle that, you're still going to hit whatever barrier that you're hitting. But if you tackle that problem, then really you can pretty much just keep going like the Energizer Bunny, I think.
I think Hallmarks of Aging's pretty much got it on the money with the DNA damage, epigenetic changes, loss of proteostasis and telomeres, and those being the four primary things, and they literally cause a cascade of secondary and tertiary effects. So things like altered signaling, which Irina Comboy is investigating, is covered by altered intracellular communication, but again, it's agitated by the fact that the epigenome has changed and favouring those pro-inflammatory cytokines. So the question is, if you adjust, like Belmont has done in this paper, if you reset that epigenome and you reset the telomeres, what does that do to the signaling environment? And I'm probably guessing it probably improves it.
Yeah.
So do we need to drill down that far is the question, or could we address these 3 or 4 primary reasons and stop the cascade happening in the first place?
Yeah, I think. Oops. All right, start screen sharing. I think we can certainly do that. I don't think we have to drill that far down to come up with meaningful therapies. But I'm also interested, from an evolutionary perspective, how this all works. And I kind of fundamentally think that, that it pretty much more or less, everything ages. I think there's got to be a right definition. I haven't looked too carefully at, hydras or other organisms that supposedly are immortal, but that might be a definitional issue, right? How do you, I'm sure that 1,000-year-old hydra doesn't have 1,000-year-old cells; they still turn over. So maybe cruder organisms. When I say cruder, I mean less differentiated cell types might have. It's more of a quasi population, but I think there is a. I think there is a fundamental evolutionarily conserved system at play that cells use to keep themselves from, entropically going into equilibrium. Because you can imagine each cell is a little micro vessel that, thermodynamics is pushing into accumulating junk, and somehow the cell has to keep ahead of that, either by secreting it out if it's not dividing, or if it's dividing, at least in yeast cells, or and certainly other cells have been looked at, shunting it off to one other cell and keeping the population alive as a whole. But yeah. Anyway, there's a lot to talk about. I don't know, Steve. I think we've wrapped up this journal club.
Yeah, if people have got suggestions for what they want us to talk about next time, I mean, I'd like to talk about the Comboy papers. I mean, I would love to take a swing at everything that Irina's been doing since 2005, and putting that into perspective. But it's what people want to listen to, what people want us to talk about, I suppose, and
Yeah.
We're always happy to talk about new stuff.
Certainly are, and there's a lot of new stuff coming out.
Too much. Who can keep up?
We're trying.
But yeah, I think we will beat that 120 year wall, and I think we'll do it by robust repairing. It's like you say, cells are little self-contained units to a certain extent. They have homeostasis, and if you can keep that homeostasis, how long do they keep trucking?
Mm-hmm.
Well, we're going to find out.
All right. Well, I guess everybody enjoy the rest of your day, and we'll be signing off. And I guess pay attention for the next journal club that'll be coming up at some time in the near future. And this is Oliver Medvedik signing off.
Yeah, thanks everybody for tuning in, and don't forget to tune in next time. And if you've got suggestions, do email them to us. And this is all possible thanks to the support of our patrons and the Heroes campaign. And every dollar counts. So if you can help us out, that would be great. Thanks for coming.